So in this note, we are going to be looking at cell division, and we're going to be looking at what it is, and we're also going to be looking at why cells do it. So let's start with just a couple facts that might help you appreciate cell division a bit more. 100 trillion cells make up your body. It all actually began as a single cell, a single fertilized egg. So you had one cell, that fertilized egg, and that actually divided into 100 trillion cells. So you can see how cell division plays a very large part in shaping you and literally what you look like. An example of how rapid cell division happens in your body, red blood cells die and are replaced at a rate of 1 million every second. So look at your clock, let 1 second pass, 1 million red blood cells within your body have died and been replaced by cell division. So cell division is very important in keeping you alive, and it happens very often. So now let's talk about why will cells divide. So cell division, we know it happens very often. Why might cells divide? There's really a couple main reasons. The first is healing and tissue repair. So your body can heal yourself. If you've ever had a cut and you form a scab, then that scab falls off and you've got nice healed skin, your body's able to heal itself through cell division. So an average human actually loses 105 pounds of dead skin in their lifetime. That's kind of gross. That's like you could build a new person out of your dead skin in a lifetime. That's actually what a lot of the dust in your bedroom is just dead skin. So that's your motivation to clean your room today. So we shed skin and other cells naturally to allow ourselves to repair the tissue. So to repair your skin, your body's going to naturally shed your skin. Another reason that you might want to divide cells is to grow. You started as a teeny tiny single cell, and now you're your large self. That's due to cell division. So if you did not have cell division, you couldn't grow, and we'd all be about one cell height high. The other reason that you might want to go through cell division is perpetuating life. So it's the way to create new organisms. You can do this two different ways. You can create new unicellular organisms. That's through fission. We're going to look at that a bit more in grade 11. You can also create gametes. So that's your sperm and your egg. That's in multicellular organisms. So that's also through a process of cell division. It's a different type of cell division though. It's called meiosis. We're going to study it in grade 11 as well too. So in grade 10, we're just going to focus on the very basic kind of cell division. And the very basic kind of cell division is going to be the cell cycle. So we're going to be taking a look at the cell cycle. Cell cycle is a continuous process. So cycle means it's something that cells are constantly going through. So I'm going to show you a diagram that shows you it's really like a giant circle. The cell is always going through these phases, and it's the actual life cycle of a cell. We're dividing that into two major phases. The first phase is called interphase. This is where the cell spends most of its time. The second phase is called the cell division phase. We also call this mitosis. So most of the time the cell is in interphase. This is not the division stage. At this stage, we're doing other things. We'll talk about that in a second. The cell division time is a very small portion of the cell cycle. So most cells at any given time are not dividing. So here we've got that diagram of the cell cycle I was talking about. Let's zoom out so you can see it. So you can see it's circular. This is representing the stage of a cell. And here we can see most of the cell is in interphase. There's 3 different stages of interphases. We'll talk about that in a second. A very small part of this is going to be mitosis and cytokinesis. This is when you're actually going to see a new cell is going to be produced. So then that same cell is going to go back into interphase. Small portion of it is mitosis, producing a new cell, and then that same cell goes back into interphase. So just because a new cell is being produced, it doesn't mean the cell is dead. It means that this cell is ready to re-enter the cell cycle through interphase. Let's look a little bit more at interphase and what happens. So this is a period of time in which cells get ready for division. So think about it. If you want to take something and you want to divide it into two of the, we call it clones, right? It's really the same version, 2 exact same versions of itself. What do you need to do? You need to do 2 things. You need to have the cell grow. So the cell needs to grow because these cells are not shrinking inside. We don't take a cell and chop it in half. The cell needs to get bigger so that way when it divides, it ends up the same size as it started. It also needs to duplicate. So it needs to duplicate the DNA, and it will often duplicate organelles to allow the other cell to be fully functional after division. So in order for a cell to divide, it needs to do 2 things to prepare. grow and duplicate the DNA, and it does both these things in interphase. So my different stages of interphase, we say G1 is called the first growth phase. So we're growing. We also produce new proteins and organelles. S is the second phase of interphase. That's called the synthesis phase. During this phase, we're actually making an extra copy of our DNA. Remember, the DNA is a blueprint for your body. It's how to build the cell. So each cell needs to have a copy of the DNA. It's very, very, very precious. We need to make a really great copy of it. And we're going to talk about that in a little bit, what happens when you make a mistake. And the third phase of interphase is called G2, or the second growth phase. This is the shortest phase. And here, cells are going to produce organelles and other structures for the new cell. Now let's talk about mitosis, which is a term you're going to hear quite often. Mitosis is a type of cell division in which the daughter cell receives the same number of chromosomes as the parent cell. So essentially, we are making clones. I used that word before. So we call this the parent cell. What's produced is called the daughter cell. The parent cell is going to have the exact same number of chromosomes as the daughter cell. So essentially, we're making identical copies of our parent and daughter here. That's the type of cell division we're going to study. There is another type of cell division, sexual cell division, meiosis. You're going to talk about it in grade 11. So the phases of cell division, the phases of mitosis are very distinct, and we actually call it IPMAT. And we throw a C at the bottom. So you have to make sure that you know these phases in order. So interphase leads up to mitosis or the cell cycle. So interphase is part of cell division. It's not technically, interphase is part of the cell cycle. It's not technically part of cell division. Cell division or mitosis is prophase, metaphase, anaphase, telophase, and telophase is followed very closely, sometimes almost simultaneously, by cytokinesis. So the entire cell cycle, IPMAT, and then the actual cell division part, just PMAT. Let's talk about prophase, and that's the first stage of cell division. So prophase is the first stage of cell division. We can actually see a real microscope picture of what prophase looks like. What happens is chromosomes, that's what contains your DNA, condense. So you can see these short, fat, like blobby lines here. Those are chromosomes. So chromosomes get very, very thick during prophase. The nuclear envelope, so that's that nuclear membrane. It's the actual membrane that holds the nucleus together. It starts to break down, and these centrioles are going to move to opposite poles. So in cell division, remember, we have to make a copy of our DNA, and then we actually have to get the DNA moved into another cell. So in order to make it so it's easier to handle the DNA, we make them really short and fat. We condense them so they're easier to grab. The nuclear membrane has to break down. So the nuclear membrane is actually what's holding the DNA in the nucleus. So if we want to have that DNA travel somewhere else, that membrane has to be broken down. And the centrioles are actually going to act to pull the DNA apart. So you want to have them at different poles. What poles means is I'm going to have one on one end of the cell and another on the other end of the cell. Let's talk a little bit more about these centrioles. So we talked about them in the cell organelle note. We said they kind of look like stars. They're small protein tubes that help to organize the movement of chromosomes during cell division. They have something called spindle fibers. Those are protein structures that guide chromosomes during cell division. And there's also something that's called a centromere. That's a structure that holds chromatids together. So essentially, centrioles are these star-like structures that are going to use spindle fibers. And the spindle fibers are going to be almost like lassos that are going to reach out, and it's going to help us to grab our DNA. Our DNA is chromosomes. Chromosomes look like this X shape, and chromosomes are two chromatids held together. So we have one strand here called a chromatid. We have another strand here called a chromatid. They come together to form this X shape, and the centromere is what holds that X shape together. So let's take a look at the difference between chromosomes and chromatid. So chromosomes, which is what we actually have forming in prophase, are tightly packed. So we say chromosomes are condensed. They are found only during division. So chromosomes begin to form at the very start of prophase, and they are not used for protein synthesis. So protein synthesis is when you actually read the DNA, read the blueprint to make proteins. Chromatin, on the other hand, are unwound DNA. They're found in interphase, and they're used for protein synthesis. So essentially, I want you to picture this. One second. So we've got our visual, we've got our ribbon here. So DNA are very, very, very long strands. So we're going to picture this as our DNA. And DNA, we can actually, when it's a very long, thin strand, we can actually read it, and it's going to help to produce proteins. So when we have chromatin, we have these long, fragile strands. That's what's found in interphase, and we're actually able to read it to help to make proteins. Now the issue is this is very, very fragile, and you don't want to damage your DNA when moving it. And that's what we're doing in mitosis, is we're moving DNA. So to make sure it doesn't get damaged, what we do is we take it and we make it so it's easier to manage. If we had to pull this apart, we're gonna get tangled, we're gonna get knots. I don't know if you've tried to like pull yarn for a long time, but you get knots really easily. So what we're gonna do is we're gonna tightly pack this. So we actually take proteins and we wind the DNA around the proteins, and it's gonna make like a thick, fat little package. So that's what a chromosome is a thick, condensed version. It's very tightly packed. It's only found during cell division to help the DNA move easier without getting tangled. The issue is that I can't read this right now. When it's wound up like this, I can't read it. I cannot make proteins. So once we've gone through and the DNA is finished moving during cell division, we unwind it again. We've got chromatin, and that's what's going to be in interphase. So in terms of a picture, this is what our chromatin looks like. We really can't see much. It's because it's so thin, whereas chromosomes look very thick. So I always like to think of this as like spaghetti, right? If you've got spaghetti, it's overcooked, it's really sticky, it sticks together, it breaks really easily, whereas I like to think of this as penne. So short and fat, doesn't stick together, nice and easy to move. After prophase, we have metaphase. So metaphase is when the chromosomes actually line up in the center axis of the cell. I like to call this the middle. So metaphase is when chromosomes actually line up in the middle. They're guided there through spindle fibers that come from the centriole. So we've got this DNA, it's going to escape from the nucleus, and then we're going to line it up in the middle. The reason that we want to line it up in the middle is we want to make sure that we have a correct copy of the DNA. So we want to get all of our chromosome 1s together. We want to get our chromosome 2s together, chromosome 3, to make sure that everything is present and to make sure that I'm going to pull the right DNA to the right side. You need to make sure you have a copy of chromosome 1 to a copy of chromosome 23. If you're missing a chromosome, you can have a birth defect, or you could have some sort of issue with your structure of your body. So you need to make sure you have a complete copy of your DNA. Otherwise, if you're missing some information, it can cause problems when you're trying to form your body in birth. After metaphase, we have anaphase. So the chromosomes in metaphase are in the middle. Once the chromosomes in metaphase are in the middle, in anaphase, they actually split apart. So I say that in anaphase, the chromosomes move apart. So they're splitting up and they're going to get pulled into their chromatids. So remember, the chromosomes were that X together. We are now going to split them up into chromatids, and they are going to move to opposite sides of the poles. So we've got one pole here, and we've got one pole here. Essentially, this pole is going to become the nucleus of cell number 1, and this pole is going to become the nucleus of cell number 2. So these chromatids are being pulled towards the new nucleus here, and these chromatids are being pulled towards these new nucleus here. It's being pulled by those spindle fibers. In telophase, a new nuclear membrane is going to start to form around each set of chromatids. So now the chromatids are no longer in the middle. They're now in their new nucleus, and you can see that they're actually starting to not be condensed anymore. So they're starting to uncondense, and they're starting to go back into chromatin. We're going to form a nucleus around here, a nuclear membrane around here, and that's going to be the new nucleus of cell number 1, and this is going to be the new nucleus of cell number 2. And right hand in hand with Telophase is the idea of cytokinesis. Cytokinesis is a division of cytoplasm. So this is actually the outside of the cell. So inside of here, we have a new nucleus forming for cell number 1 and cell number 2. The cytoplasm actually has to pinch off into our 2 separate cells. So we had one really big cell, needs to pinch down into 2 separate cells. That's what cytoplasm is, and this happens after nuclear division. So once you have a clear nucleus on this side and a clear nucleus on this side, you're going to go ahead and you're going to pinch the cell off into 2 different parts. And what do we get at the end? We are going to have 2 identical cells that form, and these cells are going to be daughter cells. The daughter cells are going to have the same number of chromosomes as the parent cell. So here we had our parent cell having 2 chromosomes. We doubled those chromosomes in the beginning. So that way when we divide into 2 cells, we've got 2 cells now called daughter cells. And these daughter cells have the exact same number of chromosomes as the parent cells here. These are called diploid cells. So mitosis always go along with diploid cells. We'll talk about haploid cells again in grade 11. So now go ahead, watch the video on mitosis. It's a little bit more interactive because you can visually see what's happening. And then please make sure that you have a note where you actually go through and describe what's happening in each phase and also what each phase looks like.